My name is Carly Brown, and I worked on a collaborative project with several other students to uncover universal signatures of life using complexity metrics. Detecting life beyond Earth is a core goal of astrobiology research, but verifying that a signal has biotic origins is extremely challenging because many biosignatures are shared between both living and non-living systems. The search for universal signatures of life, sometimes referred to as agnostic biosignatures, aims to resolve that issue. These signatures need to be characteristic to all forms of life, not present in abiotic signals, and not reliant on Earth-based assumptions. Identifying even one universal signature would be a great opportunity to focus the community's search for life in a way that our current broad base of biosignatures doesn't allow for. One of the leading examples of an agnostic biosignature is complexity, which characterizes the behavior of a system that has multiple interacting parts. Complexity is difficult to quantify with a single metric, but we can focus on applying a few specific metrics such as Shannon entropy and statistical complexity to our data to quantify features like randomness or repetition in a given time series. The eventual goal of this work is to identify complexity metrics that will help us categorize signals based on their origin. To this end, our group has applied various metrics to a broad range of time series datasets, including abiotic physical signals, biological signals, and technological signals. By plotting the values of these complexity metrics, we hope to reveal clustering within our datasets that will distinguish signals as living or non-living. My primary focus during this project has been on exploring the complexity of animal communication, specifically audios of bird vocalizations. Before we can apply the complexity metrics, we pre-process our data. To do this, we resample the original audio, which is shown in the bottom left of the figure. Then we truncate and discretize a sample of it, visualized on the right. This data is then collected in a CSV file for further analysis. We also perform a fast Fourier transform on the data, which shows us the range of frequencies present in the audio and highlights the peak frequency, shown in the upper left. I process time series data for 5 distinct bird species, including both calls and songs. Calls are typically simpler vocalizations, such as short notes or phrases used for immediate needs, such as warning nearby birds about danger. In contrast, songs are used for more complex communication, like courtship rituals, and they're often much longer, with a distinct structure and rhythm. Both calls and songs can either be vocalized by a single bird, which I've labeled as solo, or by a pair or group of birds, which I've denoted as duets. So the plot on the left denotes the bird species and the results of a first-pass complexity metric called the zip compressibility ratio. The zip compression algorithm looks for patterns and redundancies in a file and encodes them to reduce the size of that file. If a file has low complexity or many repeated patterns, then it compresses well. A highly complex file will have less effective compression. Here we define the ratio as the original file size divided by the compressed size, so a higher number actually indicates that the file had a lot of redundancy and therefore lower complexity. The figure on the right shows statistical complexity versus Shannon entropy. Higher values of statistical complexity indicate more intricate structures and patterns, while higher values of Shannon entropy indicate a greater randomness and a less predictable sequence. Essentially, data that scores high on both metrics would be highly unpredictable, while also having underlying structures and patterns that help organize the unpredictability into a complex system. There are a couple recordings that scored low on both metrics, which indicates a simple, predictable pattern with little to no complex organization. The results shown here do mirror the relative complexity shown in the zip compressibility ratio plot, which is what we would expect. Unfortunately, the time series I analyzed showed minimal clustering using these metrics. Most notably, on the rightmost figure, we can see that the 4 solo calls, which are represented by hollow circular markers, cover the full range of complexity values. For the most part, the solo songs, which are represented by solid circular markers, are slightly more clustered and show both high statistical complexity and Shannon entropy, but the trend isn't quite strong enough to draw meaningful conclusions from this data alone. There are a few immediate steps that I can take to improve the quality of my results, including better isolating the vocalizations in the audio files and comparing vocalizations based on the bird's observed behaviors. By coupling behavior to vocalization, I can hopefully see if the data spread is more a result of species variation or if it's the behavioral function that impacts call complexity. Beyond that, the real impact of this work will likely show itself when our group is able to fully compare our individual results. I plan to continue this work, and I'm looking forward to seeing the further results from our group's analysis. Thank you.